Professor Gessel, I had a quick question.
For it.
So I kind of embarrassed myself because I was just got off with Wiley Lab because I was trying to complete my cranial nerves Wiley Lab. I thought it was due tonight before midnight, but I didn't read where it stated that like it was letting me access it, but
Yeah.
it wasn't letting me answer any of the questions, and it said I already completed it when I didn't. And I contacted Wiley Lab and they were like, can you send me screenshots? And then they were like, oh, well, it's because it was due at 12 a.m. today.
I
So-
think that's what Justin was getting at the other night. But let me, you and someone else right now. So give me 1 second and I'll do that. Bear with me just for a second. Right now. Other questions, concerns? Zena, I know you're just getting on, or you're just getting on. I think I just read your email. And,
oh, it's okay.
Well, I think you're talking about the same thing that Samantha needs.
Oh,
And so.
I'm sorry.
No, it's all right. So I'm just going to do it right now.
Okay. Thank you so much. I appreciate that.
Yeah, of course.
I'm gonna go with my angel raviolos, which have to be cooked perfectly for this to work. These are some of the hardest things to make.
Any other questions or issues?
I missed what her question was. Could you repeat it?
Well, they were just looking for an extension for the exercises that were due last night. Do you need that too, Kara?
I don't believe so. Which ones were the ones that were due last night?
20 and 21.
Oh, yes, I do.
Okay. Bear with me just for a second here. Samantha, your last name, it starts with a B. Yes, okay.
I'm not sure if I turned that in yet or not.
Who? Savannah? I'll look. Give me 1 second here. I'm just gonna extend it out until the 10th, Zena, and I'm gonna do the same thing for Samantha or Sam.
Okay. Thank you.
And Kara. If I do it right now, I'll actually remember too. So Kara. Kara, I think you turned it in.
I saw my name at if you scrolled up a little bit, it's under David.
Oh, it's not under Norland. Okay. Where is it?
Or it's under, it's Kara Fleshman.
All right. And who else was that? Oh. All right, who else was it? It was Savannah. Okay. So tonight, I want to do part of chapter 14 and then get into the special census. You'll see that the special census PowerPoint is not very long because I really do want to focus on this dissection that I'm ready for. But I want to, I think I'm going to skip around a little bit with the, bear with me. with chapter 14. We stopped off, I think, on slide 87. And I'm going to skip to 105. And the reason is because I want to cover the cranial nerves with you. Because of that case study, I want to cover that right now. And then next week, we will finish chapter 14 and start and finish chapter 15. We'll finish and start chapter 15 and most likely start chapter 16. What you'll see is that they aren't very long. If I remember right, the autonomic nervous system, looking at the PowerPoint, it's 63 slides. A lot of it is a review of what you've seen before. Some of it isn't, but it's not as complicated as some of the things you've seen before. And bear with me here. And the special senses, I'm focusing what we've been told to do, and tonight's an exception to the rule because we're doing that eyeball. We're going to dissect this eyeball that I have here. But what we've been told to do as AMP 121 professors is to focus in on one special sense in particular. And the one that I'm most familiar with and that is the most comfortable for me because I know the most about it and I have the most experience with it is hearing and balance. You'll see that this is only 41 slides. And so by the end of the semester, and I'm trying to remember just how many more days we have left. By the end of the semester, we should be finished with everything. I think the muscle is a longer chapter. Looking at our syllabus and seeing how many more days we have left. So today this is the week of the 6th. So we have 4 more days. And we should very easily finish. You will have your second exam within, plan on having it next Tuesday and having it start next Monday night. I may, if you'd like me to, I'll open it this weekend. But it will cover 12 through 14. I think we also had, it's actually covering 7 too. $7:30. Yeah, I think what we had was we had it so it was covering, the first exam covered chapters 1 to 3. So this one should cover 7, 12, 13, and 14. I think I'm going to leave 15 off because I think that's a lot. So your first exam, I will open it up over the weekend and it will cover chapters 7, 12, 13, and 14. Remember, your first exam covered 1 through 3. And so this one will cover 7, 12, 13, and 14. Your third exam will cover 15, 16, and 11. And then you have a comprehensive final. Any questions?
I did have a question about the due dates for chapters 13 and 14. It's chapter 13 is showing that it's due on the 8th, and chapter 14 is showing that it's due on the 11th. And I just wanted to confirm if that was correct or not.
Yes, we did, because I had some students after class last Monday or last Wednesday ask me to push them back. That's why they're pushed back the way they are. I can push back. I'm not going to push back chapter 13 because we did finish it over a week ago. But I could push back 14.
And 15 is showing due on the 11th as
I'll
well.
push that one back
Okay.
because we haven't even covered that yet. But
Okay.
you're going to realize it's a really short chapter. The special senses and the autonomic nervous system are really short.
Okay. Thanks.
You're welcome. The muscle, I'll just show you so you see what we're looking at also. The muscle is
When did you say exam 3 is going to be due?
it will be due at the end of the semester, and you'll have a week
Okay.
because
I
it's
was
going
just
to,
trying to calculate the weeks, and I was like, wait, you said we only have 4 weeks left, and
yeah,
I was-
it will cover chapters 15, 16, and 11.
Okay.
Oh, why am I not seeing what I want? Suck. This is the 11 PowerPoint right here. You will have one more case study. And if you want me to, I will load it. I'll load it this weekend also. I'm the only one home for the next 10 days. My mom came in, she was gone, and then she left again, and now I'm by my lonesome once again. But that also gives me a lot of free time. And so I will load that, but you'll see that the chapter 11 is 97 slides. And if you think about it, the one that we're covering over the brain and the spinal cranial nerves is 97. And so I'm sorry, muscle is 97 slides. The brain and cranial nerves is 127 slides. Do you have any questions? That was a little chaotic. Do you have any questions? So let me just reiterate really quickly. And if you'd be willing to turn on your cameras, I'd appreciate it. So I'm not just looking at a bunch of names. But we will cover.
You want to come to my drug test with me?
What? All right, I don't think I was supposed to hear that. We will cover your second exam, which I will open up this weekend. Will cover chapter 7, 12, 13, and 14. Your third exam will cover chapters 15, 16, and 11. You will have one more case study, and I will post it this weekend. And so altogether, you have 3 case studies. There was one other thing that I wanted to look at really quick.
When is the second case study due?
I can't remember. It honestly doesn't matter to me. Why don't we have it due on April 20th? Does that sound all right?
Yes, thank you.
Let's see here. Oh. With regards to the lab class, got one more thing. So your fourth lab quest should be opening this week. We only covered, this is option. So we skipped over the autonomic nervous system because I chose to do that an extra day of lecture and get us caught up. We're not going to do a lab over the autonomic nervous system. I think it would be better to do a lab quest that covers the spinal and somatic reflexes, the brain structure, and the special senses. So it will open tonight. Maybe not tonight, it will open tomorrow. Let's say tomorrow.
Can I ask you, Professor Gessel, like the point of the lab quest? I feel like they're just more questions that we already do and more labeling. Like, I just, it's such a long, like, assignment that, like, I
Of the
don't,
lab quest or the lab exercises?
well, I don't know. They're starting to run together. I'm just saying it feels like we do so many repetitive.
I am asking you a lot of questions on the lab exercises.
Yeah.
I know that I am. And I'm doing it intentionally because first off, if there's repetition involved, you're going to learn it and not have to study as much later when you prepare for the lab quest. And a lot of anatomy studying is repetition. Does that make sense? I'm doing it intentionally.
It does. I just like, I'm not, this is not my first rodeo, but I'm not a traditional student. So, I just sometimes I'm like spending, so much time away from my family,
Right,
doing
right.
these different lab exams that,
Right.
so I was just asking.
Yeah. Yeah, I can understand. All right, let's jump into tonight.
I'm sorry,
When
Professor.
did we say Lab Quest 4 was going to be due? Open
It will be
tomorrow?
due.
It was number
Let's
2, right?
let's make it due in 2 weeks. I will put out an email when I put it out. I will tell you, I only finished putting together the special senses exercise probably about an hour ago. And so I haven't even chosen out the questions because I haven't done this lab in the past. I'm really doing it because Ivy asked about the eyeball. Normally I wouldn't do it at all. I think it's a good one. But normally I wouldn't be doing it at all. So I haven't, it's not something I prepared that I've just been able to fix that I made before tonight and made before the last couple days. So I haven't completely got the questions for the lab quest set up yet. That's what I'm saying. So give me until this weekend to get it done. And I will put it out and you will have at least a week to do it. Other questions? Okay. So let's jump into tonight. And like I said, I would like to go through the cranial nerves. So go through part of lecture. It will not take us very long because we've been talking about the cranial nerves off and on. And it's the last part of chapter 14. And then we will jump into the lab because you will learn that a lot of what we're going to talk about in with regards to the special senses has to do with the cranial nerves. Okay, so we ended on, we ended on 87, and I'm not going to start on 87. I'm going to start on 105. Because I want to start just with the cranial nerves. And then on when we meet again next Monday, we will finish chapter 14 and do chapter 15. So that's where I want it to be. Okay, so cranial nerves. You guys, if you've gotten a chance to look at that case study, a good thing to use for both of the case study, the best things that you can use is this part of the PowerPoint for chapter 14 and the lab PowerPoint that we did last week. So cranial nerves. There are 11 and a half to 12 pairs, and they arise from the base of the brain. They're going to exit the cranium through foramina, and they're going to lead to muscles and sense organs located mainly in the neck and head. They're ordered according to how they come off the brain from anterior to posterior. So the olfactory nerve is the first one, cranial nerve number 1. The optic nerve is cranial nerve number 2. The oculomotor nerve is cranial nerve number 3. The trochlear nerve is cranial nerve number 4. The trigeminal nerve is cranial nerve number 5. The abducens nerve is cranial nerve number 6. The facial nerve is cranial nerve number 7. The vestibulocochlear nerve is cranial nerve number 8. The cranial nerve number 9 is the glossopharyngeal nerve. Number 10 is the vagus nerve. And then remember that when it comes to their locations, the accessory and the hypoglossal switch places. So accessory is cranial nerve number 11. Hypoglossal nerve is cranial nerve number 12. And like I said the other day, if you go online, there are so many mnemonics out there for learning the cranial nerves. The main one that I use, because I'm a fan of Harry Potter.
You know,
What? What was that? I don't know. Is on they traveled and bear with me here, and found Voldemort's guarding very ancient are ancient Horcruxes. And so on the first on, I think the way that I remember the olfactory is the first one is you only have one nose and two eyes. So the first cranial nerve has to do with the nose, and that's the olfactory one. So olfactory. Optic. Oculomotor. Trochlear. Trigeminal. Abducens. Abducens. Facial. Vestibular cochlear. One thing that I will mention about the lab exercises is you have less to do now than most AMP classes have. And I recognize that it's a lot of work, but you have less assigned to you right now than the classes where I was given the material and what I gave my students last semester, which was less than it was before when I was given the material. And so I recognize it's a lot, but that's part of that is that's the way it is. Sorry, and now I'll get off that. Glossopharyngeal. Vagus. Accessory. and hypoglossal. And to know whether they're sensory, motor, or both, some say sensory, money matters, motor, but both. My motor, brother, both. Says vestibular, cochlear, sensory. Big bucks, glossopharyngeal, both. Vagus, both. Big bucks matter more. Spinal accessory is motor, hypoglossal is motor. So that's the way I keep them all straight. Are there any questions about this? So find one that works for you. Spinal nerves versus cranial nerves. So spinal nerves are designated according to the cervical vertebrae that they originate, from which they originate. Cervical 1 through 8, thoracic 1 through 12, lumbar 1 through 5, sacral 1 through 5, and the coccygeal nerve. The cranial nerves are Roman numerals. Spinal nerves, there's 31 pairs. Cranial nerves, there's 12 pairs. Spinal nerves origins, spinal cord. Cranial nerves, the brain and the brainstem. Spinal nerves, number of roots, there's one dorsal and one ventral root. Remember, the dorsal one is sensory, the ventral one is motor. For the cranial nerves, except for cranial nerve number 11, which remember has 2 roots, and we'll see that again today. They have a single root. Spinal nerves are mixed. Cranial nerves mostly are mixed, with some being sensory and some being motor only. So most motor fibers of the cranial nerves are going to begin in the nuclei of the brainstem and lead to glands and muscles. In contrast, sensory fibers, like the optic and olfactory, are going to begin in receptors located mainly in the head and neck and lead mainly to the brainstem. So cranial nerve, mnemonics for cranial nerves, I think we've gone over this a couple times, but here's some more. Olfactory nerve, the first one, is your sense of smell. Damage to it will cause an impaired sense of smell. Specifically working with the olfactory bulb and the olfactory tract. This is the only sensory cranial nerve that does not go through the vagus, excuse me, not through the vagus, through the thalamus. It's the only cranial nerve that doesn't go through the thalamus. That's part of why your sense of smell is so involved in memories, is because it doesn't go through the thalamus. I think there was a question on your case study about whether the what nerve might not be myelinated. It's the olfactory nerve. because of how it sits. It's right here. Bear with me and I'll look and I'll tell you. Case study. Okay. It isn't the way I thought it was. Which two cranial nerves are myelinated by oligodendroglia instead of Schwann cells, and are therefore more vulnerable to diseases like MS, multiple sclerosis. One of them is the olfactory nerve. So the optic nerve, sensory, involves your sense of vision, and damage to it can result in blindness in part or all of your visual field. It's also involved with regulating the shape of the lens, as we will see in a few minutes. The oculomotor nerve controls muscles that are involved in turning the eye up, down, and medially, as well as controlling iris and lens and upper lid. Damage to the oculomotor nerve can result in a drooping eyelid, dilated pupil, because it's affecting your irises. If you think about the irises of your eyes, they're regulating how much light enters your eye. And your oculomotor nerve is going to determine how much your iris is a muscle. And if it's contracted, your eyes are dilated. No, if it's contracted, your pupils are constricted. Is that right? No, I keep getting it confused. The muscles of your iris, if they are relaxed, your pupils are constricted. If they're dilated, the muscles of your iris are contracted. And that makes a lot of sense if you've ever seen or listen to books or read books like I do, where guess how they determine whether someone is dead or not? Their pupils are constricted. They're not dilated, they're constricted. Because what that means is that there's no innervation to their iris anymore. So damage, drooping eyelid, dilated pupil, double vision, difficulty focusing, inability to move eye in certain directions. Trochlear nerve is controlling the superior oblique muscle, so one of the extraocular muscles. Damage to it can cause double vision and inability to rotate the eye inferior laterally. Trigeminal nerve is the largest cranial nerve, and it has 3 branches. It's the most important sensory nerve of the face. It's got an ophthalmic, meaning your eye, so 3 coming like this. Ophthalmic is the top one, maxillary is the second one, and mandibular is the third one. The third one is both motor and sensory. The first 2 branches are sensory only. The abducens nerve controls eye movement to the lateral rectus muscle of the eye. Damage results in the inability to rotate the eye laterally at rest, and the eye rotates medially. This is the mnemonic that I use for remembering the cranial nerves and what eye muscles they innervate. So lateral rectus is cranial nerve number 6. Superior oblique is cranial nerve number 4. And all others are cranial nerve number 3. Your facial nerve is cranial nerve number 7, and it's mixed. It innervates many of your facial muscles and is involved in your facial expressions, regulating your salivary glands, nasal, and palatine glands. It's sensory in that it innervates the anterior 2/3 of your tongue. Damage to it produces facial muscles, sagging facial muscles, and disturbed sense of taste. You have no sweet or salty sensations. Bell's palsy is a paralysis disorder that affects cranial nerve number 7. Most likely caused by the herpes virus. Appears abruptly and disappears spontaneously. You have a question on the case study which says something about someone that has an acoustic neuroma. So acoustic meaning hearing. Facial nerve is coming out like this, and it has 5 branches. And they have, I think the question says that they have sagging of the face or a drooping eyelid. What cranial nerve is involved? It's a facial nerve. Justin, you're the only face I can see. Do you see that question? He's not paying attention, are you?
No, I haven't looked at the case study yet
Okay.
entirely. I just kind of skimmed it.
Oh, let me I'll tell you really quick. Yeah, Justin, now you're on in the hot seat because you're the only one that I can see at this moment. Let's see here. Okay, so question number 4. Has a right acoustic neuroma presented with right-sided facial paralysis and the inability to close her right eye. Which cranial nerve is likely affected and what may this condition be called? This is Bell's palsy and it's cranial nerve number 7.
Is that question number 4 in there, or?
What did I say? I think I said 6, but it's cranial nerve. It's question number 4.
Oh, okay.
If you go back and you look at what cranial nerve number 3 says, it gives you what we on the PowerPoint just now. It tells you the different functions besides eye muscle innervation that cranial nerve number 3 is involved in.
Okay.
Vestibulocochlear nerve, the major nerve involved in hearing and equilibrium / balance. Damage to it can produce deafness, dizziness, nausea, and loss of balance, and a nystagmus, which is involuntary rhythmic oscillation of the eyes. Vestibular because of balance, cochlear because of hearing. Actually a merging of 2 nerves, the vestibular nerve and the cochlear nerve. The glossopharyngeal nerve is involved.
Question.
Go.
If that nerve innervates the ear, how does it deal with the oscillation of the eyes? Do you know?
That's a really good question, and I was just thinking about that, and I don't.
Does it only innervate the ear, or does it also have a branch that goes to the eyes?
I don't think it has a branch that goes to the eyes. But one thing that might be involved here is it's going to affect. It is going to affect the inferior colliculi. And if you remember, they're involved in hearing reflexes.
Mm-hmm.
If you have rhythmic oscillation of the eyes, what that might be related to, having never seen it before. And if my mother was here, she would know a lot more than I do. That's a balance thing,
Hmm.
which is your vestibular cochlear nerve.
Hmm.
That's what I was just reading as well. That
What
it's
did it
due
say?
it's due to the balanced input. The balanced input from the vestibular system.
When we bring this up, think about this, because when we talk about the special senses, because I told you I picked hearing and balance. One thing he talks about, and you know, with you guys with your little tiny kids, if you've ever spun them, you know how you pick them up, you grab them by their hands and you say, let me spin you. And then you run them around and spin them a couple times, and then you put them down and say, walk straight, and they can't do it. You know, you've done it. We've all done it at some point, or watched someone do it. And if you look at their eyes, they're going like this, because even though the world's not moving, the solution in their semicircular canals here is still moving. And that's making them think that they're still moving. We will talk about it, and we'll spend a lot more time talking about it over the next couple of weeks.
Okay, really fast. Alan said he's treated this a lot, and he said that it innervates both, and that the biggest thing that causes nystagmus associated with that is benign proximal positional vertigo, which is about the crystals in the ear and the vestibular system
Otoliths.
out of balance.
The
Yeah.
otoliths, okay.
Yeah. So that is one of the big reasons why that nerve causes the nystagmus. It innervates the eye enough that when that's off balance, that's what it does. It's a whole
Is
lot.
it doing the ask him, is it doing the otoliths, or is it doing the crystal ampullaris?
Hang on.
Okay.
The crystal.
Okay, all right, that makes more sense to me. Okay, all
Yeah.
right. Tell Alan he has to listen to tonight, and he has to listen to when we go over special senses.
I will. I'll tell him. He's just out of earshot, but I'll tell him he has to.
Okay. Does that answer your question, Max? Awesome. Okay. Cranial nerve number 9. So sensation to the posterior 1/3 of your tongue. So this is another sensation of taste. So facial and glossopharyngeal are both taste. Vagus is, you know what, I'm trying to remember. I thought I heard that vagus was also involved in taste, but I'm not sure about that one. But vagus nerve, most extensive distribution of any nerve. Damage to it is fatal if both are cut, because it is your major parasympathetic nerve of your autonomic nervous system. It has a major parasympathetic role, and you can't live without a parasympathetic nervous system. If you read your book about parasympathetic versus sympathetic nervous systems, you can live without a sympathetic nervous system. According to the studies they've seen in dogs, you can live without one. You can't live without a parasympathetic. And this is the major nerve involved in the parasympathetic nervous system. And so that's why it's fatal if they're both damaged. So sensory, major role in control of cardiac, pulmonary, digestive, and urinary function. Motor, swallowing, speech, and regulation of viscera. Accessory nerve motor controls swallowing, head, neck, and shoulder movement. And damage to it can cause impaired shoulder, neck, and head movement, with the head moving towards the injured side. This is the one that I've mentioned a couple times, that technically it's not considered a cranial nerve anymore. About 20 years ago, they discovered that it's not coming off. It's not completely coming off the brainstem or the brain. It's got two branches, and if you remember, it said that cranial nerves technically only have one root. This one has two. It's got one that's coming off the brainstem, and it's got one coming off the spinal nerves, cervical 3 through 4. And it was only seen when people were dissecting a bunch of cadavers and looking at this nerve and realized that it wasn't coming off the brainstem, at least not completely. And so unlike the other 11 cranial nerves, the accessory nerve has a double origin, where the spinal portion is originating outside of the skull. Cervical nerves C1 through C5 and C6 converge to form the spinal part of the accessory nerve, and it runs superiorly to enter the cranial cavity through the foramen magnum. If you look in your book, it probably tells you, doesn't give you that much information. This citation right down here is where that information is, the journal where that was published in. So technically not a cranial nerve, because if we're looking at the cervical spinal cord, this is the second branch. This is the first branch coming off about where the medulla oblongata is. The second branch is coming off the cervical spinal cord. And glossopharyngeal nerve motor, last one, controls tongue movements involved in speech, food manipulation, and swallowing. If both are damaged, one cannot protrude their tongue. If one side is damaged, the tongue is going to deviate towards the injured side, and there's going to be ipsilateral atrophy, meaning same side atrophy. So overview. Sensory is green here. Motor is red, showing how the cranial nerves are affecting each of the different organs. Which of the cranial nerves is responsible for regulating visceral activity?
C.
It's C, right? Good job, Max. Damage to this nerve could result in defects of eye movement.
A.
It's not. Why is it not? Sam, what do you think? Say it again when you're not muted.
Oh, I was saying, I think it's the trigeminal, actually.
It's not the trigeminal.
Oh,
Is it D?
Wait, Sam, let's see. Bear with me, Kira. Let's see if Sam, what do you think it is?
Oh, it's the abducens.
Yeah, it's the abducens nerve. because the cranial nerves that are involved, optic nerve is solely sensory. So it's not going to be involved in eye movements because it's not motor. That's the reason it's not the optic nerve. And the only other nerves that are involved, I remember that mnemonic, LR6, SO4, AO3, meaning that cranial nerves 4, 6, and 3 are involved in eye movement. All of the others are not. Questions about the cranial nerves? Okay, let's jump into the lab.
Wait, I'm sorry. Could you repeat? You said 4, 6, and 3.
4, 6, and 3 are involved in eye movement, eye motor.
Awesome. Thank you.
You're welcome. So let's jump into the lab tonight. And I will tell you, I don't believe this lab is going to be as difficult for you as other labs. The PowerPoint is not as long. It's got a lot of pictures to it. And you'll see that it's just not very long. So special senses. With regards to the tongue, there are 3 types of papilla, meaning bumps on the tongue. There's valate or circumvallate papilla, fungiform papilla, filiform papilla, and foliate papilla. So these are the nipply things that you can feel on the surface of a tongue. The one that they all have taste buds on the internal surfaces in the grooves of them, except for the filiform papilla. And the filiform papilla look like little combs or brushes. If you've ever had a cat lick you and they have that sandpaper tongue, it's because their tongue is predominantly these filiform papilla. If you started grooming yourself as often as cats groom themselves, your tongues would become very filiform papilla too. Right now, they're not. But the taste buds are on the internal surfaces. And with a pore, gustavatory hair cells and receptor cells, and the sensory axon. Remember, this is predominantly glossopharyngeal and facial nerve. It's predominantly taste. Dealing with the olfactory senses, if you remember, we talked about the crubriform plate when we were talking about bony landmarkings. Crubriform plate, crubriform meaning a filter or a sieve, has a lot of little holes in it. And nerve fibers from the olfactory bulb are infiltrating these holes and moving down into the nasal cavity. if you look at these, do you remember we talked about there were 3 basic types of neurons? There were those pseudo-unipolar or axonic or something to that effect, depending on what textbook you look at. Multipolar and bipolar. Do you remember which one was specifically more common in the special senses?
Multipolar.
No, you're close, but no. Multipolar is the most common throughout. What one is very specific to the special senses? You have 2 other choices. What do you think it could be?
Bipolar?
It's a bipolar. If you look at these, actually, they don't really show them very well here. They don't really show them here, but this is one of the places where we see bipolar neurons. All right, we're shifting gears again, talking about the ear. We're going to spend most of the time tonight on the ears and the eyes. So I'm going to zoom out just a little bit so that I can see a little better what I'm looking at. With regards to the ear, there's 3 basic parts. There's the external ear, the middle ear, and the internal ear. The external ear includes the auricle, the helix, the lobule, and the external auditory canal, up to the tympanic membrane, aka the eardrum. This is the external ear. Highly innervated, very sensitive to pain. And I'll tell you this when we talk about special senses, and I'll mention it tonight also. Earwax is an immune response to keeping things from taking up residence in your ear. It smells funny and it's gross. And that is the reason bugs don't decide to live in the warm, moist environment of your ears, is it repels them. Your middle ear includes a tympanic cavity and 3 bones, the malleus, the incus, and the stapes. Malleus is attached to the tympanic membrane. The stapes is attached to the oval window that leads into the vestibule, that leads into the cochlea and the semicircular canals. The cochlea and the semicircular canals and the vestibules are part of the internal ear or the inner ear. This tube we talked, and we didn't talk about it. This auditory tube or eustachian tube is a tube that's connected from your middle ear to your nasopharynx, the back of your nasal cavity. This is why when you fly and you need to equalize the pressure in your middle ear, you yawn or you swallow. is because it's going to equalize the pressure in your middle ear with the atmospheric pressure.
I have a quick question. I don't know if it kind of piggybacks off of earlier how we were talking about when you spin around, how the nerve kind of senses that you're still spinning even though you're not. When you do like ear flushes or ear washes, how some people can have that dizziness sensation, is that part of the reason also? Because the water's swashing around and spinning around kind of in there?
It is. And you know what? For when I will tell you that I had kind of an argument with a hearing aid specialist because I do wear an aid. I don't wear one when I'm teaching online at home because I get easily distracted with it. But you have this area right in here. I mean, this is probably, and Justin's Allen can probably tell us, but this is in width and distance. The distance between this and this is probably less than a centimeter. And so these are full of a solution, a gelatin solution. And we'll talk about it here in a few slides. If you warm this up or alter this right here, you're going to alter the fluid in here. And that's what makes you dizzy. Does that make sense? Because of the kind of hearing aid I wear it completely in the canal hearing aid because it's the only one that works for my kind of loss. And so it sits about right here in my ear. And what that means is that my ear produces a lot of earwax because it thinks something is trying to take up residence in my ear. And if I let that build up too much in my ear, it will damage my hearing aid. And so I'm regularly cleaning my ears out. And a lot of times when I do that ear flushing, when I use a Debrock solution and I clean it out, and then I go to flush it out with warm water, I have to grab a hold of something or I will get dizzy and possibly lose my balance. For that reason right away there. And I've learned from experience, a lot of hearing aid specialists don't understand that because they don't understand how closely correlated these are. Does that answer your question, Sam? More than you wanted?
No, that was good. Thank you.
So the ear functions specifically in hearing and balance. Summary of hearing. So sound waves are focused by the pinna or the outer ear, and they are compression waves of air. And as they enter the ear canal, they make the tympanic membrane vibrate at that same resonance wavelength. That results in the malleus, incus, and stapes starting to vibrate at that same wavelength. As they are vibrating, the stapes is attached to the oval window, which is attached to the cochlea. And the stapes, when it's vibrating, causes the oval window to start to vibrate. And the fluid that's backing the oval window right about right here starts to vibrate, causing rhythmic vibrations in the cochlea. These rhythmic vibrations are changed into chemical messages via action potentials. and the cochlear nerve passes those electrical signals onto the brain. The semicircular canals are for balance. And the result is that we hear sound. So what we're listening, what we're essentially feeling is we're feeling compressions, we're feeling vibrations of our middle ear that starts the middle bones, the inner ear bones vibrating. And then they're pushing on fluid that's in the cochlea, forcing that fluid to vibrate at that same wavelength. And those vibrations are then translated into electrical messages via action potentials. So outer ear, pinna or auricle, that's going to direct sound waves into the ear. The external auditory canal or meatus has ceruminous, I think that it's not ceruminous, it's caruminous and sebaceous glands. Caruminous meaning earwax glands, and sebaceous meaning sweat and oil. With dead skin cells produces cerumen. My mother's tried to correct me, and now that she's not here, I can't remember how to pronounce it right, but I think it's carumen. With the guard cells, it protects the outer end of the canal. The tympanic membrane is going to vibrate as sound waves enter the ear. The middle ear, or the tympanic cavity, amplifies those sound waves, those compressions of the air, and transmits those signals to the inner ear. The malleus, so with this picture, what we're looking, here's the eardrum right here, the tympanic membrane. Here's the malleus, here's the incus, and here's the stapes. This is a neat picture because having seen what my own eardrum, when I went to one audiologist, he had a camera and he said, "Do you want to see what it looks like?" And I said, "Yes." And he showed me, and it looks like this. But the malleus, the head of it is attached to the tympanic membrane. So here's the malleus, here's the tympanic membrane. Here's the incus, and here's the stapes. So they're innervated by the vagus and the trigeminal nerves, and they're highly sensitive to pain. Why do you think your ear canal and these bones would be highly sensitive to pain?
So we can limit like damage to them.
Yeah. Yeah, I mean, usually if you feel pain, you stop doing what you're doing. The incus and the stapes looks like a horse saddle stirrup. And that's where the name comes from. The inner ear is described as a tube within a tube. It's a bony labyrinth in the temporal bone. And within that bony labyrinth of tubes, there's a membranous set of fleshy tubes. The fleshy tubes, if you look here, and I'll zoom in here a little bit. You zoom in here. Scoot over.
Jacob, it's amazing because you
If you look here, here's the bony tube, bony labyrinth. Here's the membranous labyrinth. So the membranous labyrinth is within the bony labyrinth. The membranous labyrinth is filled with endolymph, and the bony labyrinth is filled with perilymph. The endolymph is similar to intracellular fluid in its consistency. The perilymph is similar to cerebrospinal fluid. And so this is the fluid that when that stapes starts vibrating against this oval window here. Here's the oval window. Here's the stapes that would be attached right here. When that stape starts vibrating on this, it's going to cause rhythmic vibrations of this fluid, just like splashing in a pool. And so the receptors within the ear are going to be sensitive to those vibrations and convert them to electrical signals and action potentials. So the cochlea, so middle ear, outer ear, cochlea, and the semicircular canals attached to the vestibule. The cochlea is the organ of hearing. The vestibule and the semicircular ducts are organs for equilibrium and balance. And this is all within the temporal bone, that bony labyrinth. This is one of the pictures that your manual asks you to label. So here's the answers right here. So right here is the oval window where the stapes would be attached. Here's the cochlea. Here's the vestibule and the semicircular canals. And like I said, the vestibulocochlear nerve is a merging of two nerves. The cochlear nerve going to the cochlea and being involved in hearing. The vestibular nerve being involved in balance. So the scala media is the cochlear duct. If we do a cross section through this cochlea, remember what cochlea means? We talked about it when we talked about the nasal cavity, the bones of the nasal skull. Those, what were they? The middle nasal cochlea, superior nasal cochlea. You remember these? Maybe. Meaning they're shell-like in shape. That's where the cochlea gets its name also. It's shell-like in shape. So there's three chambers here with the cochlea. There's the scala tympani, the scala media, and the scala vestibuli. The cochlear duct, the scala media, is the middle chamber, and it's filled with that endolymph. It's separated from the scala vestibuli by the vestibular membrane, and separated from the scala tympani by the basilar membrane. It contains, within this cochlear duct, it contains the spiral organ or the organ of Corti, which is the acoustic organ. So this is where the vibrations of the liquid and the fluid is going to be converted into nerve impulses or action potentials. So looking at it, this same picture, probably this right here, maybe this right here, a microscopic cross section of it. I have to remember this here. Scala tympani, scala media, and scala vestibuli. That scala media is also called the cochlear duct. Lining the very bottom of the scala media is that spiral organ of Corti. Right here. And so this is just an enlargement of this area right here. So here's the basilar membrane that's separating the scala tympani from the cochlear duct. And up here would be the scala vestibuli. And this is the scala media or the cochlear duct. And right here is the organ of Corti. That includes hair cells. So the organ of Corti has an epithelium that's composed of hair cells. These hair cells are receptors. They have long, stiff microvilli called stereocilia. And there's a tectorial gelatinous membrane that sits across the top of them. So if you look right here, I'm trying to enlarge it a little bit so you can see what I'm seeing. Right here, here's the basilar membrane. Down here's the scala tympani. This is the cochlear duct. There are hair cells. There's 3 rows of outer hair cells and 1 row of inner hair cells. And then there's a tectorial membrane that moves across the top. This is filled with perilymph, endolymph, endolymph, perilymph. Surely, is that right? Yeah, I think that's right. So the tectorial membrane is sitting on top of those hair cells. That's going to be important in a moment. But that membrane is sitting on top of those hair cells right here. So there's 4 rows of hair cells. There's 3 rows of outer hair cells. 3 rows of outer hair cells that adjust the response of the cochlea to different frequencies. So this is involved in cochlear tuning, increasing the precision of sound. The inner hair cells is a single row and it provides for the actual hearing. So here's the hair cells, the outer hair cells, the inner hair cells, it's tectorial membrane, the basilar membrane. Vibration of those ossicles in the middle ear is going to cause pressure waves in the inner ear fluid via that stapes that's attached to the oval window, which is attached to the cochlea. And the result is that when those vibrations start happening, boom, 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 the basilar membrane is going to start to vibrate also. It's going to start boom, 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 boom. And every time that it bumps, those cilia are going to bang up against this tectorial membrane and get opened up. They're mechanical channels, mechanically gated channels, where as they bump up against this membrane, they get open up and allow ions in. So the hair cells are moving with the basilar membrane, bumping up against it. They are these hair cells, the stereocilia of the hair cells are bathed in a high potassium fluid. The endolymph is high in potassium, creating this electrochemical gradient where the outside of the cell is positive 80 millivolts and the inside is minus 40. So this is the membrane potential of these cells. And the tips are embedded in that tectorial membrane. So the stereocilium on the inner hair cells are also mechanically gated. And there's a tip link where the top, there's 3 rows within one hair cell. And when the top one opens, the second one opens and the third one opens. So the top one, when it gets bent over, it's going to open up the second one, which is going to open up the first one. Causing a series of action potentials. These are mechanically gated channels, just like we talked about the neurotransmitter, that it binds to a channel and it opens that channel and allows ions in. This is happening by just pulling on the channel gates, opening the channel gate and allowing potassium in. As it bangs up against the tectorial membrane. So as the tallest stereocilium is going to get bent when the basilar membrane rises up, when it rises up against this tectorial membrane. Questions? as the tallest stereocilium is bent towards the tectorial membrane, it pulls on the tip links and opens ion channels. Potassium is going to flow in, and the depolarization is going to cause release of neurotransmitter, stimulating sensory dendrites and creating action potentials in the cochlear nerve. So as it gets bent, the tallest stereocilia gets bent, it pulls on the second one, which pulls on the next one, which pulls on the next one, opening up all the channels and allowing potassium to come in. So this is a quick summary of what we've been talking about here. Sound waves enter the ear and cause the tympanic membrane to vibrate. The tympanic membrane vibration moves the auditory ossicles and sound waves are amplified. The stapes at the oval window is going to generate pressure waves in the perilymph within the scala vestibuli, this layer right here. Pressure waves cause the vestibular membrane to move, resulting in a pressure wave formation of the endolymph within the cochlear duct and displacement of a region of the basilar membrane. The hair cells in the spiral organ are distorted, initiating a nerve signal in the cochlear branch of cranial nerve number 8. The remaining pressure waves will then be transferred to the scala tympani and exit the inner ear via the round window down here. Any questions? It's a quick summary of hearing. This is just showing those vibrations again, vibrations against the oval window, causing bump vibrations of the basilar membrane, which then bends those hair cells towards the tectorial membrane, causing a series of action potentials. Now talking about balance. The vestibule and the semicircular canals their major job is to work with balance and equilibrium. When sensing tilting, the vestibule, this part right here, is the major important part. It contains a macula which has receptors that are embedded within an otolith fluid. Oto meaning ear, lith meaning stone. So these are little ear stones. They look little crystals like Justin Zallen was talking about. Otoliths meaning ear crystals. The semicircular canals, on the other hand, rather than tilting and linear acceleration, they sense rotation when you're spinning or going around a corner. The receptors here are cristae receptors or cristilla ampullaris. So within the vestibule, within the macula of the vestibule, we have hair cell receptors again that are attached to the vestibular branch of the cranial nerve number 8. And they are embedded within a gelatinous, like a jello-like membrane that's got little odorless stones in it. And this gelatinous stone-embedded membrane can slide back and forth on these receptors. And when it slides back and forth, just like we saw with the hair cell receptors and stereocilia in the cochlea, these are also capable of, when the otoliths shift back and forth, when they shift back and forth, they're going to bend these stereocilia and open them up, causing a series of action potentials. So when the head is tilted, the heavy otolithic membrane sags, bending the stereocilia and stimulating the hair cells. When you do this, it pushes forward, bending those stereocilia as the membrane sags and bends up against those stereocilia, opening them up. With linear acceleration, where all of a sudden you're in your car and you hit the gas and you start going really fast and you're like, whoa. The otolithic membrane lags behind, bending the stereocilia. Causing a series of action potentials. So it's a membrane that's moving back and forth on these hair cell receptors. And as it moves back and forth on the hair cell receptors, it's causing this head tilting and opening up the different stereocilia. Just like jello sliding along a lumpy surface. It opens up those stereocilia. Do you have any questions about this or comments? With regards to that nystagmus, this is the semicircular canals, or what would, according to Justin's Allen, this would be the semicircular canals. Because rather than having a membrane, a gelatinous membrane that moves back and forth along the cell receptors, hair cell receptors, we have a cupula, which has a bunch of hair cells within it. And as the fluid moves by these cupula, it bends them back and forth, causing those hair cells to open up again with mechanically gated channels, initiating a series of action potentials. So the receptors in the semicircular, in the vestibule are otolithic receptors. Vestibular macula densa otolithic receptors. in the semicircular canals, their cupula, with crista ampullaris being the major receptor. So the ducts are stimulated by rotations of the semicircular canals, which are in the three different planes. You have an anterior semicircular canal, a lateral semicircular canal, and a posterior semicircular canal. So rotation in any of the three planes is going to alter and cause a series of action potentials in the crista ampullaris associated with each of these ducts. So each duct is filled with this endolymph that's going to open up into a dilated sac where the cupula are. And as it bends them, as the fluid rushes over it, it bends it back and forth, causing a series of action potentials. Each ampulla contains a crystal ampullaris, and the hair cells with the stereocilia are, and a kinocilium is buried in a mound of gelatinous membrane called the cupula. So as the fluid moves back and forth over it, that's where you're getting that feeling of dizziness. As it's pushed over, it stimulates the hair cells within it. Questions? Let's move into the eye. So the eye, looking at the outer eye, protective structures, the eyelids, the cavity bones, the eyelashes, the eyebrows, or to keep sweat out of your eyes, hopefully. Conjunctiva, which allow for the production of mucus and tears to lubricate your eyes. And lacrimal glands, which are going to produce tears that remove debris and lubricate the sclera. Other lacrimal structures, lacrimal meaning tears. Lacrimal gland is up in the upper lateral corner of the eye. And they secrete tears that, as you blink, cover the surface of your eye. The lacrimal canals are going to drain this fluid, the tear fluid, into the lacrimal sacs, which are connected to the nasolacrimal duct, which drains into your nose, into the inferior medial concha, meatus, inferior nasal concha. So this is why when people cry, their nose warms too. This is because the tears are running down the nasolacrimal duct into the nose. The muscles of the eye are attached to the sclera, the white part of the eye, the extraocular muscles. Looking from a lateral view, Levator palpebra meaning eyelid. So levator palpebra superioris is going to be the muscle that's involved in elevating your upper eyelid. Right inferior to it is a superior rectus muscle. Rectus meaning straight. So the muscle that moves straight across the top surface of the eye. Off to the side and moving through a pulley and attaching onto the lateral surface of the eye is a superior oblique muscle. You can see it better right here. So here's the medial surface. Superior oblique is going to move through this little pulley and insert on this part of on the lateral surface of the top, lateral superior surface of the eye. The lateral rectus muscle moving on the side and moving straight. The medial rectus muscle moving midline straight. The inferior rectus muscle moving straight down and on the bottom. And the inferior oblique muscle is like the superior oblique muscle in that it's going to come down, it's going to come down on the medial side. It's going to go through a pulley over on the medial side of the eye and insert on the lateral surface. So oblique always means that it's coming in at an angle. And so the mnemonic to remember these, which cranial nerves affect, control which muscles, is that LR6, SO4, AO3. LR6, lateral rectus, is controlled by cranial nerve number 6, the abducens nerve. SO4, superior oblique muscle, is controlled by cranial nerve number 4, the trochlear muscle nerve. All others, medial rectus, superior rectus, inferior rectus, and inferior oblique, are all cranial nerve number 3.
Can I share a quick anecdote?
Go.
Pertaining to that that picture there, there was a time that I was shadowing doctors because I wanted to be a doctor, and one of the doctors I shadowed was a pediatric ophthalmologist.
Yeah.
And he was a surgeon, and the procedure that I saw, he was correcting in children, like the eyes either going outwards like this or inwards like this.
So lazy eye.
Exactly, like kind of, but like your eyes are just like they're not going straight. They're like going out in weird directions.
Okay.
What I saw the surgeon do was sever the lateral rectus muscle and reattach it in a different place so that it wasn't pulling the eye so much to the outside. And that's how they fixed that.
Wow.
Yeah.
Okay.
Delicate procedure.
Yeah, that would be. And I've heard of that being done. Wow. Yeah, that's a sense. Wow. Yeah. That's neat. Yeah, I couldn't see how that would work though. So this is a picture, and I'm mainly including it because it's a figure from your questions. But the conjunctival fold up here, the palpebra conjunctiva. So the conjunctiva are what are lining and allowing secretions of mucus and tears. So conjunctival fold, palpebra conjunctiva, meaning on the eyelid, the inner part of the eyelid, and the cornea, meaning the outer part of the eye, outer part of the front anterior part of the eye. Another picture we're looking at about right here. The lens, the shape of which is controlled by the ciliary bodies, which are controlled by cranial nerve number 3, the oculomotor nerve. This right here, the iris, the cornea, the first thing that was ever transplanted, and the easiest thing to transplant. Why do you think it's the easiest thing to transplant?
I would think it's because it's less likely to be rejected.
Why?
By the body. I don't know why.
It's you're right. You're right. It's avascular. And that's what makes it so it's least likely to be rejected. It has very few blood vessels involved with it. Right in front of the lens, so anterior to the lens and deep to the cornea, is the anterior cavity. According to your book, your manual, this is what they call this. This is full of what's called aqueous humor. Bear with me here. We will see this. Well, we may not see that in particular, but we will see aqueous humor. That's not right either. That isn't right either. Humor. It might just be OR. Aqueous humor is anterior to lens and deep to the cornea. So that's where it's located. Deep to cornea. So fills the anterior cavity. So it's located anterior to the lens and deep to cornea. The vitreous humor is located deep posterior to the lens and fills the rest of the eye cavity. So I'm just going to say it's located posterior to the lens in the eye cavity. So this would be located in that anterior cavity, aqueous humor. The vitreous humor is in the posterior cavity. So aqueous humor here, vitreous humor here. The outer layer, on the outer layer, continuous with the cornea, is the sclera, which is the white of the eye, which is where those extraocular muscles are attaching, like the one that Max's pediatric ophthalmologist was working with. They're attaching onto the sclera to move the eye in certain directions. So the cornea is a fibrous coat that covers the iris and helps focus light rays onto the retina, and is, I'm going to leave it like that. The aqueous humor is similar to cerebrospinal fluid. Remember, it's what's located right here. The crystalline lens is going to work to bend and refract light. The vitreous body is full of vitreous humor, or it's also called the posterior cavity, according to your manual, and that maintains the shape of the eyeball and keeps the retina attached to the choroid, which is, we'll talk about those layers in one moment. So the lens works to refract the wavelengths of light to focus them on the back of your eyeball on what's called the fovea centralis, where you have the highest density of photoreceptors. If you are nearsighted, what that means is that the light is converging in front of the fovea centralis, so anterior to the fovea centralis. If you're farsighted, it means that the light is focusing to a point that's posterior to the fovea centralis, so somewhere back here, rather than right on the fovea centralis. And corrective lenses will adjust for the strength of your eye lenses. If you're nearsighted, you wear concave lenses more likely, that are going to work to help your lens adjust it to a certain, adjust those light rays so that they will focus right on the fovea centralis. And if you're farsighted, it's going to focus them right. It's going to allow you to focus them on more anterior on the fovea centralis rather than back behind the fovea centralis. Different tunics of the eye. Did we talk about tunics before? We talked about tunics. Tunics are, I've told you I like to read, and I read all sorts of stuff. If you ever have read Charles Dickens or Jane Austen, they talk about people putting on their tunics or putting on their coats, putting on their tops. So the tunics of the eye are 3 different coverings. There's the sclera, which is associated with the white of the eye, which is a dense tissue layer that covers the entire eyeball and is continuous with the cornea. The choroid, that is a highly vascularized tissue that nourishes the outer layer of the retina. And the retina receives light that the lens, receives light that the lens is focused and converts that light into neural signals, sending the signals to the brain. So from the outside in, it's the sclera, choroid, and retina. This is just a neat picture showing the optic disc, which is where the cranial nerve number 2, the optic nerve, is going to enter the eye. And it's showing the different tunics. Here's the sclera. Here's the choroid, looks very dark. Here's the choroid. And here's the retina. The retina is the one that has the photoreceptors in it. At the same place where the optic nerve and the vessels are going to enter the eye, there are no photoreceptors. So this is what we call the blind spot. Where the nerve is entering the eye and the blood vessels are entering the eye, there's no space for photoreceptors. So cornea. So this is another picture, and I'm a lot of these pictures, not this one, but this one and a lot of these other pictures, I'm including them here because they're from your manual and I've given the answers to the questions. So when you're answering those questions, use these pictures because I'm giving them to you. So the cornea, here's a cross section. Here's the cornea. Here's the iris. Here's the lens. Here's the pupil, or what we also call the iris aperture. Aperture because it's an opening into the eye. What are you seeing when you look into someone's pupil? Max, you can't answer this one because I know you know a lot about the eye now. What do you see when you look into someone's pupil?
You're looking in at their lens.
You're looking at their lens, but you're actually looking through their lens because their lens is clear. What else do you notice how it looks very black? When you look in someone's pupil, it looks really black. Because what you're actually seeing is the back of their eye. And so you're looking through this aperture, you're looking through the lens like Elizabeth said, and you're seeing the back of their eye. So the ciliary bodies are going to be involved in controlling the shape of the lens, little muscle ligaments, the choroid layer and the sclera, meaning the most outer layer, the retina being the most inner layer, and the choroid being the middle layer. And the optic disc is where the optic nerve is going to enter the eye and the blood vessels. So the optic disc is your blind spot on the retina, where ganglion cell axons exit the retina to form the optic nerve. And the retinal arteries and veins are entering and exiting the retina. And they lack the photoreceptors. You just don't have space. That's the way I think of it, is you're right here. You have this big nerve coming in, and when we look at the eye, you'll see it's a huge nerve. It's a big nerve. You have this big nerve coming in and all these blood vessels coming in, and there's really just no space there for receptors. And that's why you don't have them there. So some more pictures looking at the macula luta, which I'm trying to remember what the macula luta is. It's right next to the fovea centralis. And I cannot remember exactly what it is. Oh, you know what? I'm just going to Google it on my phone. Just like, I'm going to tell you, because I don't like not being able to tell you when I think it's something easy that I could Google in a hurry. Max, do you know what it is?
I don't, but if I had a guess, it would be like where your cones are concentrated.
Okay.
I don't know though.
So this is what it says. It is the part of the retina that's responsible for sharp, detailed central vision. It's the macula luta, also called the fovea, so I think it's the same as the fovea centralis. Contains a very high concentration of cones, so photoreceptors, and these are the light sensitive cells. This is another picture that came right from your manual. The central fossa. I looked that up before class because I wanted to know, well, what's different between that and the central fovea? The central fossa, remember what a fossa is? It's just an indentation. So the central fossa is the location of the central fovea. And that's why the macula lutea would be right on top of it. One thing I'll mention here, actually I'll mention it after this. So the retina has two major layers. It has a neural layer and a pigmented layer. The neural layer contains the photoreceptors, the rods and the cones, and the connecting neurons, which contains those bipolar neurons. So this is the other place. So we have them in our sight. I think we have them in our hearing, and we have them in our smell, bipolar neurons. The pigmented layer is highly sensitive to light, and in animals, it contains the tapetum lucidum. This is what you see when you are driving along in the middle of the night and a deer comes in front of your car and their eyes reflect back the lights of your car. What you're seeing is the tapetum lucidum in their eyes reflecting the light of your car lights back. This is what you see with your dogs and with your cats in the middle of the night. It's a solid black layer, and it allows them to just see better at night. But their vision is more blurry because you want to be able to get rid of some of that light to sharpen up your images. But it does allow them to see better at night. It's also why my one cat that you've seen a couple times, he's my firecracker. If I get him mad, his eyes get really dilated, and if I get him right in the right angle, I can see where his blind spot is, and I can see all the blood vessels in the back of his eye, because the light is being reflected back from the tapetum lucidum out at me. So if you have a cat that gets really cantankerous like mine does, I'm sure it's genetic from me. But if you have a cat that's like that and you can get them really cantankerous, if you get them to dilate their eyes, you can see all the blood vessels in the back of their eyes. Kind of fun. He doesn't think it's fun when I make him mad, but I think it's fun. So look out from the outer edge of the retina in. The pigmented layer, a darkened layer. The photoreceptors, the rods and the cones. The bipolar neurons. Ganglions, remember ganglions meaning cell clusters. And the optic nerve fibers. So there's a neural portion and a pigmented portion of the retina. Three major, this is just another picture that I know it's coming from your manual, and that's partly why I included it. But showing those three tunics, the sclera, the choroid, the pigmented epithelium of the retina, the photoreceptors, the cones and the rods, the bipolar cells, neurons, and the ganglion cell layer. And last slide, just showing pathways of sight to the brain. Retina, photoreceptors and neurons in the retina process the stimulus from the incoming light. Axons from the optic nerve from the retinal ganglion cells exit the eye. They cross over in the optic chiasm. And the optic tracts contain axons from both eyes, and these axons will project to either the superior colliculi or the lateral geniculate nucleus. Remember, the superior colliculi was part of that corpora quadrumana that's involved in visual reflexes. The lateral geniculate nucleus of the thalamus, superior colliculi, and then the primary visual cortex that's in the occipital lobe. Any questions? Okay, to save some time, I got all my dissection stuff all on all my instruments set up before class started. So I'm just going to switch cameras, tie up my hair, and put on a lab coat and open this up. So bear with me just for a couple seconds here. Oh, not that one. Okay. So this is a cow eyeball. All right, where did I just put those scissors? There they are.
Professor, can you take us out of the PowerPoint so we can see better?
Oh, yeah. I'm sorry. Thank you for
That's
saying
okay.
something.
Okay.
You can also.
Do you see it now? Is
Yeah.
it better now?
Yes.
So like I said, I get these through a company that's located in North Carolina. And they're called Carolina Biological Supply. I think the school also has them, but it's just rather than trying to go out to a campus, it's easier for me just to order them on my own. But so here's looking at it there. Here's the cornea. Cornea. The iris. What I first do, and anytime I'm looking at an organ, I try to orient myself first and look for things that I know I can see really easily. Turning it around on the back, this little area right here, that's your optic nerve. And so I'm going to, because your best dissection tool is always your fingers, I'm going to try first just to tear away some of this tissue. Because a lot of times if you start in with your scalpel blade, you will tear through something that you're going to try to find later. And then you won't be able to find it. I'm guessing that a lot of what I'm carrying through right here is sclera. Max, feel free to correct me if I'm wrong. It's really, really strong. And I don't want to flip it up into my face because that would be gross. But, all right, I'm going to cut. And these scissors are not the best ones for this. I'm going to use a scalpel.
Oh, that's just nasty.
It's not nasty. It's cool.
Are you at home doing this, or are you in a lab?
Yeah, I'm at home.
Oh.
That's why I was saying that I order all this stuff, and it's pretty easy to get. It's just like ordering on eBay. All I do is say, this is what I want. For my my nieces and nephews, they are homeschooled kids. And so one thing that we did this last summer was I went down and saw them, and we did a bunch of frog dissections, ordering them through this same company. And I learned that there's some major differences between us and frogs. Okay. I'm going to quit trying to cut through all this because I'm not cutting through it very well. And I don't honestly care that much about back here. Okay. So one thing that they tell you to do when you first start dissecting an eyeball is you want to just start cutting around the outer edge. And you want to, and remember, so we're looking right here, right to the inner surface here would be the lens. What's right behind the lens? Right posterior to the lens. Do you remember what was right posterior to the lens?
Is the aqueous humor?
That's anterior.
Vitreous humor?
Vitreous humor. So one thing that I want to be really careful of when it comes to the eyeballs, and with Lauren saying this was gross, is if I'm not really careful cutting here, it's going to squirt out at me. And so I'm going to be really careful, and I'm just going to cut right here. You can see it is kind of bubbling right around there. Hold on. Don't nail me because I'll scream. Okay. If you can you see the fluid? Okay, Justin, Ivy better be watching. Is she watching?
I'm watching.
Okay, good.
Cut it in half.
Cut it in. There, did you see how hard it was for me to cut it? Some people's children. Wow. But do you see the fluid? So what we're looking at right there, that's the cornea. This layer right here.
Okay, that's cool, but also disgusting at the same time.
It's not gross, Lauren. It's not gross. Right behind that, and I don't think I've ever seen it like this. But right behind this, if you see the white right there, that's the lens. This right here, the black right around right here, is the iris. And right deep to it would be the ciliary bodies that are controlling the width of the iris. I don't know if I can cut it in half. I was struggling just to even tear through some of it. We'll see. So yeah, if you look at this, you can see that it's kind of striated because it's essentially a circular muscle. That as it changes in width, just like your iris when your pupils constrict, as it constricts, it's going to extend in length like any other muscle. And when it dilates, it relaxes. Now let's see here.
Is it supposed to be black like that?
Yeah.
Is it black like that? It's not black like that for human beings, is it?
I don't think so. What color are a cow's irises?
I don't know. I never really pay attention. I always call them hamburgers.
They're black.
They're black. Okay, thank you. Because I'm like, no, I am not. It's been a long time since I really looked at a cow. But yeah, so yeah, it would be black. I don't know what it would be normally. You know, usually we don't, when you look at a cadaver in a lab, I don't know if I've ever paid attention to what color their irises were. So I'm going to try to do what Ivy asked and make a cut. We'll see what happens. But you can see that there's a lot of fluid around it.
Ivy did some research on a human pupil and a human iris in death.
Okay.
It completely relaxes after death and results in a fully dilated pupil with no visible iris at all.
A fully dilated pupil?
Uh-huh.
So what that means is that in comparison to other muscles, that when they relax, they become big, they extend out in length. Pupils are going to constrict. They're going to become smaller in length. Yeah, I'll have to think about that one. But I think I've heard that too. Have to think about how that occurs. So I'm still dealing with a lot of fluid, a lot of that vitreous fluid, because if you look here, I'm still pushing a lot of it out. So I'm gonna, all right, that was kind of nasty. All right, so I'm really tearing through some of this muscle. Okay. Oh, why can't I just get rid of that? What I'm trying to do is get rid of some of this just flash.
So what would cause cataracts? Would that be like an infection in the eye?
Cataracts are actually what it is it's oxidative damage to the lens. And I don't know exactly how that works. But I can tell you, you know, my little teaching assistant that I often have on my lap, she's developing cataracts. And my mom has a homeopathic thing that she does for specifically looking at antioxidation. And what we're trying to do is coat her eyes with this every morning. And she's so trusting that she lets us do whatever. And see if we can slow that down, because cataracts are oxidative damage. But right here, I don't know how well you can see that, but that's muscle. Whether I'm looking at the top or back of the eye, I have no idea. But this right here, this has to be muscle. just the way it's shaped, that has to be muscle. That's attached to the sclera right here.
It looks kind of thin. And like it looks kind of like a sheath almost.
Yeah, but you know, muscles around the eye, they're not very thick. They're actually pretty thin little muscles. It doesn't take a lot for muscles around your eye to move. We'll talk about, in a couple of weeks, we'll talk about motor units. Your eyes are actually really small motor units, meaning they don't have a lot of muscle fibers in them, in comparison to other muscles like your biceps. Okay. All right, there we go. Now we're making some headway. So right there, you can see there's your lens. This right here, all of this right here is your iris. And when we're looking at it, that's what we're seeing right inside is the lens.
So because of the eye aren't because the eyes are not as well protected as other organs, is that why like eye damage is so easy? Like because there's less protecting.
Say it
less
one more
protection?
time. Eye damage is so easy.
Like is damaging your eyes easier than damaging other organs because of the lack of protection that they have?
Well, yeah, I mean, they're really sensitive. I don't know if you've ever heard that if someone tries to attack you, go for their eyes. Because they're pretty unprotected. But when it comes to the one thing that you have with regards to your immune system is you have a lot of immune cells and lysozymes specifically in your tears that are protecting your eyes. So this that I've got in my hand right here, that's the lens.
That's super cool. I am just sitting here mesmerized.
Awesome. 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 So all I did was pull it out from behind this iris. And that looks very oxidized to me. But you can see, I mean, it's actually, this is not, this is actually really, really firm. I'm not a really strong person, but it's really, 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 really firm.
So you're saying that like the lens in our eyes should be more clear?
I think I think your lens is a lot more flexible than this is, because part of this is that it's oxidized. It's also been fixed.
Okay.
If we got one like that brain tissue that I had, if we had it like that, I don't think it would be like this. I don't think it would be because this is almost rock hard in the center.
Is it fluid filled?
Not, this is honestly, this is like rock.
Oh, wow.
It's more like a hard gelatin, a hardened gelatin. But one thing that I wanted, let's see if I can get to this and not macerate it. Pull that out. All right, let's see here. maybe. Trying really hard to actually pull out the iris. And I don't know if I can do it. If you look back here, this is vitreous humor. What I'm saying, let's see here. Oh, I lost my blade. Come here. Oh. Bear with me just for a second. I lost my blade. How, Mary, don't get yourself. All right. I'm going to try coming down this way. Okay, Ivy, I actually did finally get it kind of open. All right, so this whole little globular stuff, that's vitreous humor.
Hey, are you able to bring that back up to the camera? I'm
Yeah.
sending a picture to my sister.
Okay.
Sorry.
Nope, let me see if I can't really, it's not very easy to hold on to, but it's right there.
Is that what makes up the vitreous body?
Yes.
Ooh.
Yes.
Okay, I got it.
So I'm trying right here. Let's see. I'm going to cut right here. I'm trying really hard. I want to get out the iris without destroying it. And I'm not doing a very good job.
Do you know what causes our irises to be different colors? I mean, I know it's genetic, but
I know that it's pigment.
okay, it's pigment that causes the different coloring.
Uh-huh. Okay. Oh, yuck. Oh, sorry, guys. Okay, just flicking some stuff. So if you look on the very back, so this is the back side of the iris. And I don't know how well you can see that, but do you see the little striations there?
Yeah, we can see it.
So that's the muscle. So that's what Ivy was saying when we're looking at this, and it relaxes, it lengthens out, it becomes longer in width when the pupil becomes constricted, and becomes thinner when it dilates.
Eye color is melanin.
Yep, you're right. You're right. It's the same that makes some people darker than other people. But is it what makes the difference? Here, Ivy, here's your next question. What makes the difference between blue and green ice? Like there's certain people who have like gray eyes, there's certain people who have like blue eyes, and certain people who have like green eyes. How does that, what's going on with them? That I don't think it's all melanin, I think it's something else. So one thing that I wanted to show you on the back of this is, do you see the translucence?
Does that kind of reflect some of the light back?
Yeah, that's the tapetum lucidum.
We don't really have that. That's in the other area.
I don't think we do.
That's why I like cats and dogs' eyes glow.
I'm pretty sure we do. That's why in some
We
like
do?
flash photos we have red eye.
But do we have it to the same extent?
Maybe not, but I from what I've heard, it's different in all different animals.
Okay.
We only have brown and red pigment in our eye.
So what causes blue eyes and green
So,
eyes?
so from what Ivy's been reading is that it's the different amount of pigment and connective tissue in the iris. If you have very little, very little melanin and more collagen tissue, the eyes appear blue, like
All right.
light blue.
Like him. He
Okay.
has-
Does your dad have blue eyes?
Yeah, you just can't see it because of
No, I can't see it.
shadow. from
But
his-
that's why he's more sensitive to getting his eyes injured. And that's why he's also more sensitive to cataracts long term. So make sure he wears sunglasses. You got to make him wear sunglasses. So this is that pigmented layer of the retina. And let's see, I wanted to see, now that I've I've cut it in half like Ivy wanted. Let's see if we can find, oh yeah. Well, is that it? Sec. I'm gonna.
It would be so dark.
Yeah, it's really dark because you want, when light shines in your eyes, you want a dark black layer to absorb any light that is gonna not get absorbed so that you can see sharply. So right here, here's the optic nerve right here. And so I'm kind of going right on the very back other side of it, right next to in the inner part of the eye. That spot right here, this right here, where this connective tissue
That's
is.
the blind spot.
Booyah! Yes, that's a blind spot. Good job. That's a blind spot. You guys, is there anything that you want me to try to find?
You said you saw the, but are there any other, I guess that piece doesn't have any of the other nerves that are around it, do they?
That sclera is really tough. I didn't realize just how tough it was. I've watched a lot of students dissect eyeballs, but it's been a long time since I dissected one. And I didn't realize how I didn't realize how tough this actually is around here.
Yeah, I did a fresh one before and everything just ruptured on me.
It did it? Was
Yeah.
this tough though? Was,
Huh?
was this tough right here, the sclera?
It was tough. But then once I got through that, everything just ruptured and it was just like thicker, kind of like fluid, just runny. And it was
The
like,
vitreous, yeah.
yeah, it was.
Did you hear that I was trying to be really careful not to poke it and have it spray up into my face?
Yeah.
Yeah.
The fixed one is nicer though for this purpose.
Well, I can say that the one thing that I don't like about dissecting eyes from cadavers is breaking the eye cranial cavity to get at the eye. Because there's very few things that make me squeamish when it comes to cadavers, but that does. Because they smack it with a hammer.
Ooh.
And it sounds like a bone crunching. It's nasty.
But they don't feel it.
I know they don't feel it, but it still sounds awful. It sounds so awful. I mean, it makes me cringe. I mean, I think that watching someone saw the skull, the top of the skull off of a cadaver also just does it to me. I'm just like, blah, blah.
Oh, I think I can get so into that.
It's really cool though. If we have a place that's down in, I want to say it's in Baltimore. We have a cadaver center in Baltimore. And they actually have done tours up until the pandemic. And I had a student at Stevenson who worked there for a long time. And I'll look it up. I'll try to find out more about it and I'll tell you more about it. But they really, what they do is I'm trying to think if they use anatomical forensics or what they're doing, or if they're looking at investigating body parts. I don't know what their purpose is, but they do tours and they do allow you to go in and look and see what's going on.
Ivy, field trip.
So side note, we are going, everyone in class, the Real Bodies exhibit is coming to Reading in May, at the end of May, and we'll be there all summer long.
Really?
It's the preserved cadaver, like full exhibit. It'll be there from May 22nd to September 18th.
Gisha, you gotta come.
Where is that gonna be in Reading?
Yeah, where's Reading? I've heard of it, but
that's gonna be in Reading, at the Reading Public Museum.
I'm excited.
That's awesome. Well, I'm teaching a 121 course over the summer. We may have to take a field trip.
Are you teaching 122?
I don't know. I'm not teaching it over the summer. And they're changing. There's not gonna be a lot of online courses for A&P next year. There's some, but they're trying to push them more to online. I mean, push them more face to face. The one that I'm teaching, because I live here in Hanover, the 121 that I'm teaching is going to be at the Gettysburg campus. And I haven't heard what I'll be teaching in the fall. Are you, Lauren, are you thinking about taking 122 over the summer still?
Yeah, I'm gonna try to take it over the summer. If you guys, I noticed that Hack is pretty much already full for their remote. So like,
Have
I
you,
thought of,
do they have an online class?
they do.
Have you
So
contacted that professor?
I have.
What did they say?
So because of the hearing disability, it would not be recommended that I take AAP2 in person. So I have to try to get to like a remote, something like this.
Have you, is there a 122 that's online over the summer?
Yeah, there's there's 2 classes left that has openings.
Well, you know what, the one thing, and all right, famous last words, because when I teach online classes, there's no cap. No class gets, you can have as many students as you want in the class. Like normally they cap them at 24.
Yeah.
My 122 started at 26. Because there's no cap, because you don't have to worry about how many students you have, people you have in one classroom. So I would contact the professors. Who are the professors?
Let me, I would have to look, but I've been going back and forth with the financial aid department to get this hold removed so I can get in those classes. So let me see. It's biology.
Have you heard of the rare eye disease keratoconus? So my husband has that, and it is it's pretty rare from what the eye doctors tell him, but it's the cornea. Yeah, the cornea starts to bulge, and he has to wear hard contacts to keep it from getting worse.
Wow.
That, and his vision is poor, but that's because of it. But yeah, I think it has something to do with the collagen in the.
It's not the aqueous humor behind.
No, it has to do with the, let me see, the collagen in the eye. I'm pretty sure that they think anyway. They really don't know what causes it. But it can get to the point where, like, if you don't like wear the hard contacts to help correct it, or keep it from worsening, like your eye, like looking at your eye from the side, which is typically, cone shaped slightly, it actually will start to bulge out like pretty far. Shush, I'm talking.
Wow. Wow.
So yeah, stop it.
Puppy.
So yeah, it'll actually start to bulge out pretty far. And hard contacts they're not fun, because God forbid, first off, they're smaller than your typical contact. And if he can just be sitting there and blink the wrong way and it'll just fall right out. And you don't get a whole pack of them like you do like a throwaway contact.
No.
Nope, you get a pair. And I don't know how many times we've been in the car and it'll pop out, and next thing you know, we're pulling over and he's getting the contact off the floor of the car, putting it in his mouth to clean it off, and then putting it back in his eye, which is absolutely disgusting.
Yeah,
but you don't have, you don't have much, I guess, option in a situation like that per se.
No.
He's blinked, they've fallen out onto the ground, and then in looking for them, because his vision is so poor, in looking for them, he has stepped on them. It's interesting, but
they're expensive.
They're very
I
expensive.
used to work for Blue Cross Blue Shield Massachusetts, and their like claims department, and I know like I
They're
saw.
typically not covered by insurance.
Oh, well, BCBS MA covers it because it's required under the Massachusetts law. And I remember we would get charges for the distributors for like $1,000, $2,000.
Mhm.
And like we would cover like 25 or 50%. But it was
Yeah.
just like,
Yeah, there's a lot of insurances that don't cover them. Because it's a medical condition, though, his are covered. He still pays a couple $100 for them, but
Wow.
it's better than 1,000 or more.
Wow. Is it genetic, or it has to be genetic? Is it
So
genetic?
it's not, well, I don't know, because they say that it could start. It doesn't really start. His didn't start until like his teens, like
Okay.
early teens.
Okay. Wow. So what is
Yeah,
it
and they
called?
don't, there's not keratoconus.
Keratoconus.
And there's really no, like, they've been studying it for years. There's no real, I should say, like, unknown predispositions. Like, it can be present at birth. It could develop a little bit later, like once you hit like puberty ages.
Wow. Wow.
he should look into studies, like joining studies on like when they do them. So I know
He
like
won't.
he won't.
He won't. No, he won't do it. He's a
'Cause I
stubborn
know-
man.
Because I know like Mass Eye and Ear, they would always reach out to people and be like, hey, were you diagnosed with keratoconus? Like, do you have keratoconus?
It may,
Like,
even though, like, when you look it up and research on it, there's nothing I've come across that there's nothing definite about it. So to say it's genetic or hereditary or whatever, they don't know. But it does make me wonder, because his aunt, his mom's sister, she does have it too. They're the only two in the family that have it.
Wow.
So it does make you wonder, like, if it could be
It sounds genetic.
Yeah.
So now tell me one more time, who has it in his family?
His mom's sister.
Okay.
So his aunt.
So it's an autosomal recessive. Wow. Wow. So ultimately, what's going to happen?
As long as he continues to wear those hard contacts, I mean, you could, if you let it go uncontrolled and don't try to help it, I mean, it could cause really like painful swelling in your eyes.
Well, and the damage to your optic nerve with the swelling is bad. It's not, I mean, it's like glaucoma.
So looking at one of the government websites that I usually find it on, it does say in most cases keratoconus is not inherited and occurs in individuals with no family history of the disorder. But then it says the condition can occur in families. But you're right when you said on actually, yeah, it says inherited in an autosomal dominant pattern.
Autosomal dominant.
Yeah.
What that means is, how many kids do you have, Marissa?
How many kids do I have?
Yeah.
2.
When they say autosomal dominant, that means that 1 out of your 2 kids is going to develop it too.
Shush.
That's the chance.
Yes, it says means 1 copy of the altered gene in each cell is sufficient to cause the disorder.
Right. How old are your kids?
19 and 13.
Wow.
My daughter's worn glasses since she's oh my gosh, I don't even, 3rd or 4th grade.
Wow.
Hey, can you turn the shooting game down for a second? Thank you. Sorry.
That's all right.
That's the 13-year-old.
Autosomal dominant, but they've only seen it. That makes me wonder, because usually autosomal dominant, you see them a lot more often than one aunt in the family.
Mm-hmm.
Autosomal dominant is usually 1 out of every 2 people.
Yeah, his entire family, like both of his parents wear glasses, his sister wears glasses. Grandparents wear glasses, like everybody in the family seems to wear glasses, but not that that necessarily means anything. But him and his aunt are the only 2 that have that.
Well, I wonder.
And I've never ever heard of it in my life until meeting him.
Huh. Well, it also makes me wonder if there's multiple genes playing into it and not just one.
Mm-hmm.
Because when there's multiple genes playing into it, then, I have a good friend. I had a good, not really a friend. Once high school was over, I didn't really know him, but he was an incredible athlete. Never smoked, never drank. Ended up being recruited into the Red Sox out of high school, and died of lung cancer 2 years ago.
Who
And
was your friend?
what?
Who was your friend? Maybe I've heard of him.
His name was Aaron Thatcher. And he was an incredible athlete in high school. He played basketball, he played baseball. I know that he never drank, he never smoked. Between he and his wife, though, they had 3 kids with cystic fibrosis, which makes me think, how many precursor genes are there for lung cancer? Because, for someone who came from a small farm town, because that's where I know he grew up, in the same place where I did, where air pollution is not a problem. How do you
Did his
develop
family
lung cancer?
Did his family smoke? Because a
What?
lot of times,
He did not smoke. I will tell you, just FYI, I'm LDS, so I'm a Latter-day Saint, I'm a Mormon. And he was also, and we don't smoke and we don't drink.
So his parents weren't smokers either?
No, they were not. And so it was really shocking to me when I'm 48 now and he's 46 and dies of lung cancer. And in my head, I'm trying to figure out, well, how in the world did he die of lung cancer?
Well, I know, from what my pulmonologist has told me, because a few years ago, I was diagnosed with tuberculosis and I had to go to Mass General, and I was in isolation for months.
Wow.
Not fun. I know when I was just kind of talking to my pulmonologist about it, and he's like, about lung damage, and he said